Okay, so this is chapter 1, part 1. There'll be 2 parts for this chapter. And I hope you guys that this chapter kind of does pull some stuff together from the previous chapter, since we've been learning about cells. And we're almost done learning specifically about cells and going to get to some of the bigger stuff like genetics, real sin. So this chapter is mostly concentrated on eukaryotic cells. These are cells that have a nucleus. So cell division is more complicated because you have to make a new nucleus or divide one nucleus into 2 nuclei. So it is really complex, like I said. This over here, this picture is a eukaryote. Remember, we got our single-celled protista kingdom. And so you can see it in late stages here. It's about ready to pinch apart. You actually can't see what's going on inside with mitosis, but you can see, do you remember that other part of cell division? Cytokinesis. Awesome. Would you guys remember what cyto means? Cell, yeah. Okay, so multicellular organisms like us, we develop from a single fertilized egg, right? So the sperm fertilizes the egg, then it has all the chromosomes it needs. Cell division is also important for repair when something gets damaged, or if the cells just get old and wear out and need to be replaced. All eukaryotes have the same process. It's almost exactly the same. There are some subtle differences between the plant cells and the animal cells. I'm hoping you guys kind of remember that a little bit from the lab. And again, we call the 2 new cells daughter cells. So first, let's concentrate on the cell cycle. So there are a lot of vocab, but a lot of it, they overlap each other. There's a lot of redundancy. So looking for the relationship between the words is going to be really helpful. So the cell cycle, when you think of the cell cycle, do you picture an image? Do you picture something maybe you saw in the reading? Do you remember that pie chart, like a pie graph? Okay, so I want you to start thinking about that, because then that'll help you remember this. So the cell cycle, the daughter cells, as soon as they're formed, they're in gap 1. They're going to grow here. And they're going to do their job here. If there's a need for them to divide, then they move to S. What's S stand for? The synthesis of DNA, right? So now it's copying all its DNA. Then it will move to G2, gap 2, where it's going to get even bigger before it becomes 2 cells. So before it gets split in half, it needs to make more organelles, it needs to get larger. Then it hits mitosis. And then do you guys remember the phases of mitosis? What's the acronym? Do you guys, anybody remember that? PMAT, yeah. So let's do it. Prophase, metaphase, anaphase, telophase. Awesome. Now telophase is overlapping with what we mentioned a few minutes ago. Cytokinesis, yeah. So the PMAT, which stands for what phase? Mitosis, is specifically talking about forming 2 nuclei. So it's the nuclear division. Cytokinesis is more specific on the cell actually splitting. So lots of times people say it's the splitting of the cytoplasm. And then both of those processes make up cell division, right? Cell division. Okay? All right, so we had PMAT going on here in the M phase, and then cytokinesis, and then there's this other one, the G0. And G0 will happen, a continuation from G1. If the cell never needs to divide, they say it arrests, and they call that the G0 stage, where it's just never going to divide. Now you have a lot of cells in your body, one particular type of cell that is famous for not dividing. It can't fix itself, it can't replace itself. The eye, believe it or not, can have you guys ever had just your eye scratched?
Huh?
Well, nobody, you guys, who's had their eye scratched? Does it heal pretty darn quick? Yeah, the eye is amazing that way. It's painful when you hurt your eye, but most of the time, it'll fix itself. Now, the back of the eye has something more like I'm looking for that could cause permanent damage. The nerve cells. So what do you think of when we do that? Where?
I think of my spine.
Okay, so we got nerves going everywhere, yes. The nerves going down the spine, and then what about what's in here? The brain is all nerve cell. Well, it's an organ, so there's a couple different types of cell types, but it's mostly nerves, right? So these guys are famous that if they get damaged, they won't divide and replace that damaged tissue. There's a lot of new cool research on this, and that's what the stem cells are being used for, and they are getting some successes of helping people that are getting paralyzed. Why isn't what?
Interface.
Anaphase?
Interface.
Oh, great question. Okay, so let's look at our chart so we have a nice visual. Let's listen to this first, and then I'll point that. That's a great question, so here we go.
Eukaryotic cells are always in one of the three phases of the cell cycle: interphase, mitosis, or cytokinesis. During interphase, the gap stages G1 and G2 occur where cells grow and synthesize RNA, proteins, and other macromolecules. A cell either prepares for the next S or M phase, or performs the cell's special function in a multicellular organism. The G0 stage is a stopping point within G1. Most cells in adult multicellular organisms are in G0. When a cell in G0 or G1 receives a signal that stimulates it to divide, it passes through the restriction point, R. This point of no return commits the cell to a full round of the cell cycle. After passing the restriction point, a cell enters the S phase, during which the DNA of each chromosome replicates to form a new identical set of chromosomes. Cells pass from the S phase to G2. During G2, the cell prepares for mitosis by synthesizing specific types of RNA and proteins, which will be needed in the upcoming mitosis, or M phase. During G2, the chromosomes spread out and fill up the nucleus. Mitosis is sometimes called nuclear division, because the nucleus divides into two nuclei with identical sets of chromosomes. After mitosis, the whole cell divides. Division of the whole cell is called cytokinesis. After cytokinesis, each daughter cell enters G1. They may then enter G0 or commit to another round of the cell cycle.
Okay, so where is interphase? Do you guys see it? It's where the cell normally is, right? And do you have evidence to support that when you think back of what you saw on the microscope, where most of the cells in interphase? Yeah, okay, so it spends most of its time in interphase. So the reason that this whole phase is known as interphase is because to our eyes, it looks exactly the same. Right? So you say, yeah, that was an interphase. You have no way of determining if it's in G1 or S or G2 with your eyes and a light microscope. Is that making some sense, you guys? Okay. So when we say PMAT, we're only talking about what phase of the cell cycle? Mitosis. So, but the whole cell cycle is made up of interphase and PMAT. So there's kind of like this level. Do you guys see that? If we're just talking about the cell cycle and how the cell looks, we have interphase and PMAT. Okay? And people get hung up on that. So for example, I could define interphase as being G1, S, and G2. Do you guys see that? Okay, like we could define interphase by the cell is just doing what its job is. It's just living and doing its job. Another type of question could be that in what phase is DNA copied? Now that could be two answers. So what is it? Interphase or S, depending on how specific you're getting, okay? Or what the options are in the multiple choice question, right? Okay? Okay, so restriction points. So what they have figured out, actually let me back up real quick though. When you guys look at these circles, these cell models here, what phase are they demonstrating right here? Can you guys remember? It's way oversimplified because there's only one chromosome in our example here. But what is this one? Okay, you can see cytokinesis going on. It's just what you just said. It's what? It's anaphase, because you can still see the chromosomes. They've just been dragged. They're dragged by the centromere, so the legs of the chromosome are dragging behind. Okay, so that's why they get that V sort of shape, because the spindle fibers are pulling them right to the opposite sides of the cell. So that's anaphase. Other questions that show higher level thinking, you guys, is talking about how much DNA is inside the cell. So in what phases here, S, G2, M, G1, is there double the normal amount of DNA? It starts where? S. And in this picture, can you see there's double the amount of DNA? Okay, and it's being held together by that centromere. Even here, if it hasn't pinched in two, we have double the amount of DNA. So we have the double amount of DNA all through, whenever it got finished in S, all the way in G2, all the way through mitosis until cytokinesis has happened. And sometimes, this is type of mutation, sometimes cytokinesis will happen before mitosis has finished. Sometimes mitosis will happen and no cytokinesis will happen. So what does that leave us with? A cell with two nuclei. It needs to split in half. And if it doesn't, then it's probably just going to die because you have too much DNA in there. Okay, so what regulates this cell cycle is it's done chemically. And we have found during times, before things are about to happen, right after things have happened, like doubling the DNA, for example, there are these checkpoints. And the cell will actually arrest, the cell will stop dividing if it doesn't pass its checkpoint. So for example here, this restriction point, if the cell is going, it gets some sort of stimulus, some trigger that it needs to divide, it won't divide until it's like checked itself out. Is it a healthy cell? Does it have mutations? Does it have everything it needs? Is it a good thing to divide? If not, it won't divide. It's the same thing. There's another restriction point here in the S phase. If the DNA didn't double itself, copy itself, it'll also stop. So it's just really amazing that the cell can regulate itself so well, and most of the time, it turns out just perfect, and it makes the cells it needs. I think we went through that. And we went through that and that. Okay, so now we got to get a little more specific on DNA structure. Oh, speaking of DNA structure, is anybody going to do the DNA model? Let's do next class. Sweet, okay. Again, make sure you follow that paper and do the right thing, right strand. Make sure you make your opposite side. Okay, see me if you have any questions. So what's so neat is the structure of DNA is amazing. I mean, I think if you just look at it, what a crazy cool looking molecule. But then how it copies itself is just amazing. And it's all because of the base pairing. A's always bond to, and C's always bond to. So if this thing was to split in half, the enzymes can easily rebuild the opposite sides. And that's how it replicates. It doesn't start from scratch. It's not like if you see here on the board, that's kind of a messenger RNA. But if we had this whole piece right here, it's not like the chemicals look at this and make a whole other set of DNA. It happens through this replication process. I'll show you. We went through that. Oh, let me show you something before I move through this. It's an anti-parallel, which means one side goes in one direction and the other side goes in the other direction. Do you guys see that by the model? Look at this, the way the sugars are pointing, that pentagon shape. Do you guys see that? Okay, and so it's anti-parallel like that, which is important when it comes to replication because the enzyme will only work in one direction. The other thing that's really cool to look at is they're shaped. T's and C's are shaped very similarly, whereas A's and G's are two rings. Do you see that? And they fit just perfectly together. And actually, look at the difference between how they fit together, the C's and the G's versus the A's and the T's. What's different with how they're bonded together? Got it? Right, you guys are both exactly right. So which one's stronger then? The what? The G's and the C's are stronger, and this makes sense actually in looking at bacteria that live in hot springs. They have more C's and G's. They live in a very tough environment where the DNA can unzip or these hydrogen bonds can break more easily. So it makes sense for their survival that they would have more G's and C's. And they do. So this molecule needs to unzip or these hydrogen bonds need to break for two reasons. One, to get the DNA to copy itself, and then the other reason is for it to make the proteins off of it because it's our genetic blueprint. So yeah, let's think about that. So covalent bonds are very strong bonds that go through chemical reactions for them to happen. Hydrogen bonds are just like the water, how it aligns and it's attracted to each other. So it's a very weak bond. Yes, good analogy, okay? And when this is being built, the enzyme does need to help the covalent bonding happening, and it also, this happens pretty easy where they're just attracted to each other. Okay, so what happens? So for DNA to copy itself, first we have an enzyme, and the enzyme's called DNA polymerase. Do you hear the ACE at the end, like all enzymes, catalase, amylase? So it's a big enzyme that is going to help the reactions happen. So first it attaches to the area where it's going to unwind the double helix, which there's other hydrogen bonds that are holding this helix shape also. It's not, otherwise it would just be like a flat ladder. Go for it, Cole. But so these, it'll unwind it too. Then the third step is the synthesis of a new matching strand off of the existing strand. So we call this the replication origin wherever it starts. And again, I mentioned that enzyme. So the last little part, you know how we always then try and talk about the little guy, right? The prokaryotes, they don't have a nucleus, so it's not nearly as complex, but they still got to copy their chromosome. And then they have cell walls like the plants. So then the spindle fibers still grow and just separate that one chromosome, right, into two because it had copied. And it's called binary fission, which is simply splitting in two is what that means. So here's the cool picture. Now, remember the anti-parallel thing I said. The polymerase, that enzyme, only fits in one direction. So it can only go one direction. So look at this closely. So if I imagine a zipper, right? If you're unzipping a zipper, you're unzipping it on one side. As the hydrogen bonds are breaking, the polymerase just moves down, building the opposite or the complementary side as it keeps getting open and open and open. But the other side needs to be built in the opposite directions. So the zipper has to get far enough down for the polymerase to be able to attach where it just opened, and then it chugs along going this way. And then it pops off, and then it's gone down and it comes again, and so we're building it opposite. But because it takes up space to attach, and then it pops off, there's these openings. So it's built in fragments. Another DNA polymerase, now that there's plenty of room, another one can come in and fix those. Okay? So one side is called the leading strand that's going just fine. Lagging, the one that has to wait for it to open. And we're going to do a model, and you guys will try and model this correctly. Okay, making sure you understand the relationship between chromosomes and DNA, this double helix. So if you look at this picture real closely, the DNA is over here and it's, in its double helix. And what happens, it starts coiling, spiraling upon itself. Then there's these histones, these 4 proteins, they bundle in 4. These proteins that help it package it even tighter and wind it tighter. Then that thing starts to coil on itself. And so all this is happening in prophase. So we have all this loose DNA, and it starts supercoiling into these long chromosomes, these long pieces of DNA, linear. And here is one side held together by the other side. This is one regular full chromosome, and here's its exact copy. And I think of that centromere as like a paper clip, like keeping the copies together. Let's stop. I think I went a little bit more. Okay, so then the other thing to think about is when the DNA copies itself. If the wrong base is put there, then that's a mutation. And it happens, but it's extremely rare. The rate of mutation is like 1 in a million of those base pairs. But you have billions of base pairs. So then you can get these mutations. And mutations are not always bad. Mutations are what makes us different. It also gives us new traits that could be beneficial. But lots of them also are bad. So the second bullet, this is important. When we get to how the gene is made, how the protein is made, the mutation might not cause a problem at all. And that's called a silent mutation. Go in there in those boxes. You got to get one of each. And it looked like there's showy in the bottom one where there shouldn't be. Okay, so what I was saying is a base could change, but because of the code, it might still code for the same amino acid. Don't worry about that now. We'll go over it soon. Well, so one strand of DNA has just millions of them. And most genes, well, hold on, let's get to it later. The codon is, you need 3 amino acids for one particular amino acid. And then it depends on how long the protein is for how long the gene is. So wait on that one. Yeah. Okay, so diseases like sickle cell anemia would be a bad mutation. It messes up the hemoglobin shape and it doesn't work as well. But things like hair color, depending on where you live, that mutation of a blonde hair of not making the melanin, not making the dark pigment. Melanin's actually on your skin. I'm not sure what the name of the one in the hair is. It might not matter at all. And then as you'll see when we get to the codons, it might not change the protein at all. So hang out on that. That's going to be our next chapter. Okay, let's see here. The other thing to consider is we get mutations all the time in our body cells, and normally they're not a problem. If anything, they don't divide and they arrest and they don't move on. But it's all about our environment that we're in if we screw up our DNA. But if we screw up our sperm or our eggs, then we can pass these mutations on to the next generation, right? Or if we're born with a particular mutation, we can definitely pass it back. So the chemicals that cause mutations in the DNA are called mutagens. You might be familiar with carcinogens also, and that's a type of mutagen that causes cancer. Not all mutations cause cancer. Okay, the other thing that's amazing is there's also proofreading. So the DNA's been made, another DNA polymerase will come along and go, and if it can't fly right down the DNA, like if something's sticking out weird, it'll notice it and it'll stop and it'll proof it. It'll edit it. So it's just kind of amazing. That's called excision repair, where it cuts out the wrong nucleotide and replaces it with a new one. Okay, and so that's where we're going to stop.